Okay, it's 9 o'clock, so let's get started. Today we transition from sort of our first module, which was getting introduced to R, how to use R for plotting, data exploration, and a little bit of statistics, although we didn't really do too much. What we're going to move into now over the next probably couple months, few months maybe, is an introduction to concepts and probability. And I think this is a really important aspect of science in the sense that, in many ways, every scientific discipline uses probability to help understand the phenomenon that are being studied. And that's really because what we're dealing with is uncertainty. Basically, everything that we study or that's worth studying or is interesting to study has uncertainty associated with it. If there was no uncertainty, we wouldn't really be interested in studying it. We're trying to understand why and when and how and under what conditions different phenomena happen. And so I really like this quote. This comes from a series of articles that are listed here at the bottom. And it I think, kind of reflects the difference between probability and statistics. And we'll touch upon that over the next couple of lectures. I think probability and statistics sort of get intertwined, but they're different. Statistics we use really not to tell us whether we're right. We're trying to model the uncertainty about whether or not we're wrong. And the underpinnings of statistical methods are really probability. So probability is really the mathematical or conceptual framework that statistical methods leverage to help you understand the probability or the chance or the likelihood that you're wrong. I'm going to revisit this link at the end of the lecture, but this comes from a series of articles whose name I always forget. I have it written at the end. But there's probably 20 short 1 to 2 page articles about different concepts in statistics and probability that are really quite nice. Okay, so really probability is about modeling uncertainty. We know there's uncertainty in biological processes. There's uncertainty in a lot of things that we do day to day in our own lives. And I think one of the real challenges in teaching probability and actually thinking about probability is humans, I think, are just psychologically or logically not terribly well equipped to think about uncertainty. And I think one of the key examples of this is somehow we get influenced by past events and think that the past somehow will predict the future. And many processes that happen in our lives and also in biology are totally random and totally independent of the past. But yet we sort of build past experience into our psyche, and that impacts the way we think about future probabilities. So some things that sort of illustrate humans' issues with uncertainty in the future. You know, we've got these things like the magic 8 ball where you have no idea and you sort of roll this thing and it'll tell you yes, no, or I like this one, don't ask me, I'm just a ball. We can read tea leaves, and if you've seen the movie Big, you probably remember Zoltar. This is this thing that's put a quarter in and it tells you your future. So I think all of these sort of silly things that we use day in, day out, maybe for fun, reflect our challenges with uncertainty. But more germane to what we're talking about in this group is the uncertainty associated with biological processes. And I think one of the fundamental things we need to do in each area of our research, especially as a PhD student or a postdoc, is really be able to think deeply about the uncertainty associated with the biological process, the experimental techniques we're using, and the way those processes and techniques can vary from day to day, month to month. And so I've been in meetings with some of you, and I know some of you know Jay Gertz. Jay Gertz has a really nice question that he always asks PhD students, which I think is great, is sort of like, what's happening to the molecule? Imagine you're a molecule that you're studying. Like what are the things that can impact the fate of that molecule? And that reflects sort of the uncertainty about biological processes. There are many different things that can impact the fate of an RNA molecule, whether it becomes a fully mature protein that can do something in a cell, for example. So some examples of uncertainty in biological processes include, how many mutations exist in a cell division? Well, we have an estimate of the expected number of mutations, but that's really just an average. That gives us essentially the probability that a mutation will occur, and then we propagate that across the entire cell when the genome is replicated. But the actual number of mutations that exist in one cell division or another cell division can vary dramatically. Which actual alleles do we inherit from our parents? That is also a random process, or mostly random, and therefore there's uncertainty. Where do crossovers actually exist in a meiosis? We know that there's crossover events. We know where there's hot spots and where there's cold spots for recombination, but the actual crossover sites and therefore the actual haplotypes that you inherit from your parents, that's an uncertain process. How much time does it take to get from the expression or the transcription of an RNA molecule to becoming a folded protein? And for instance, in the context of RNA-seq, how many molecules from a given gene or a given allele are observed from a single cell or from a collection of cells? That is a uncertain process. Okay, we also know about uncertainty from sampling. So probability helps us model the uncertainty of biological processes. But another really useful application of probability is the fact that it's often far too expensive to do an assay on the entire population of things that we're studying. So maybe we would really like to be able to look at every single cell in the liver of a mouse, right? Well, that's really, really expensive. So what we end up doing is taking samples, smaller samples that are either easier to access, that might be one reason, or it's just cheaper to do the study. Maybe you want to study gene expression in the liver, so you just take little punches, and you hope that assay, that small puncture that you took, or that small sample, is representative of the larger population of cells. And I think, as we know from the last election, that these samples, seemingly random samples, can often belie the truth. So let's follow up on this example of studying all the cells in a given tissue or a given organism. Let's imagine that's way too expensive, because often it is. So instead, what we want to do is take small samples, and we might be smart and realize sort of that this is a bit of an uncertain process, and so instead of taking just one sample, we'll take many small samples, and then the idea is we'll combine those many small samples, which are still nowhere near the entire population. But let's say we take 3 independent small samples, and our hope is that maybe that first sample didn't totally represent the entire population, but the collection of the 3 different samples is a much better approximation of the entire population. That's the hope. So let's imagine we take these green circles are actually gumballs, but let's imagine they're cells that represent different clonal populations. So each of these different colors have distinct mutations in them that sort of characterize one cell from another. So the first sample, we get some red cells, we get some green cells, and we get one yellow cell. The second sample, we actually don't get any green cells this time. We just get more yellow cells. We get some new cells, some orange ones, and blue. And now, okay, this is kind of the intuition. Maybe that first sample had bias. This second sample has bias, but together they represent a better approximation of the entire population. And now we take this third sample, which again is finding entirely new cells. And we would think, we can do a lot of what goes into these designing experiments like this is thinking about how many samples, what sample size we need to make sure that we have a good approximation of all the cells that are present in a given tissue. 3 is often used. But what if the truth is actually something like this, where it's a very diverse population, but the actual frequency of each of these different clones is essentially the same. But from those 3 different samples, our observation is that some of the cells, colored gumballs, are more abundant than others. But the truth is that they're all relatively equal in frequency. And this is where sampling, we need to understand that uncertainty about sampling, the degree to which it actually reflects the entire system that we're studying. And probability theory really helps us think about whether the degree to which that's true, or model that uncertainty. Okay, so over the next couple of lectures, we're going to be introducing some terminology and some syntax for thinking and writing down ideas about probability. And we're going to talk about, especially when Alan Rogers takes over for the next 2 lectures, we're going to talk about 2 different schools of thought for probability. And you may have heard of, who's heard of Bayesian probability theory? Okay. Who's heard of frequentist? Okay. So they're related, but they're sort of distinct ways of thinking about probability. And there's some nuances. I've come up with some basic definitions of this, but there's some nuances to this, so this isn't completely representative of all the nuances and distinctions between frequentist and Bayesian. But the frequentist school of thought is that probability reflects the relative frequency that a given thing occurs over the long term. So what does that mean? Let's say under exactly the same conditions, we measured a phenomenon 1,000 times, and the conditions were absolutely identical. And let's say 700 of those 1,000 times, the event that we're studying occurred. So the frequentist school of thought would say, well, the probability of that event occurring is 0.7. 700 times out of 1,000, and the 1,000 is representative over the long term. We've measured this thing over and over again, so we can sort of build up some confidence in the pattern that we're seeing is actually real. So, for instance, if you get on your weather app, what does iPhone say? Like on every hour, it'll say there's a 70%, for instance, chance of raining. What does that actually mean? I think really that's a frequentist measurement. It's saying that one way of thinking about it is a frequentist measurement, saying whenever we see the conditions that we predict will occur at 4 p.m. this Friday, rain happens, right? But 30% of the time it doesn't. In contrast, the Bayesian way of thinking is really a degree of confidence or certainty about a phenomenon. And that's, as Alan will talk about in maybe a month or 2 weeks, that degree of certainty or degree of belief is taking into account alternative possibilities. So it's making a prediction given one possibility and many other possibilities. And essentially that probability that comes out, that confidence or the degree of belief, is essentially taking into account all these alternative possibilities. Where, in contrast, the frequentist approach is really just thinking about, well, how often does something occur when you've done many, many experiments over the long run. We're going to be revisiting these distinctions over the next few lectures, so if it's not entirely clear, bear with us, we'll revisit these things. The next 2 lectures today and the next one are really going to be focusing on this first concept of probability, the frequentist notion of probability. Okay, so our goals today are to develop a basic intuition for probability, understand one type of discrete random variables. We're going to talk more about what discrete random variables are and how they're useful in biology. Just very quickly, most of what we do with things like ChIP-seq and RNA-seq and DNA-seq, They're essentially counting experiments where we put a bunch of molecules from a bunch of cells into a sequencer. The sequencer gives us a bunch of reads. We align those reads back to the reference genome in some way that's relevant to the question that we're asking, and then essentially we count things. And those counts tell us something about the biological processes that were happening in the cells that we studied, and those counts are discrete random variables. Discrete is essentially just saying there's integer numbers. There's either one read or two reads. There's never 2.1 or 2.1735542, right? It's always integers. We're going to talk about Bernoulli trials, the binomial distribution, sampling, the law of big numbers. We're going to talk about using simulation in R. We're going to revisit R, and we're going to talk about how we can use simulation to do really kind of cool experiments with a few lines of code that demonstrate the probability theory of some discrete random variables. And then if we have time, I'm hoping to touch briefly on this notion called the gambler's fallacy. Okay, so probability, as I've already said, this is really useful in the context that there really is uncertainty in the biological processes we study, and it's on us when we interpret our results and we model our results and we write conclusions about our results. It is our duty to understand the probability of the uncertainty associated with those measurements, right? We're never right. It's never the truth, right? It's a model of what might be the truth. So an example of this, very simple one, is Punnett squares from, gosh, I guess for me it was high school, it's probably middle school these days. But let's imagine we've got mom's genotype on top at a given position in the genome. She's heterozygous AG, so she inherited A from her dad and G from her mom, say. And dad is heterozygous AG as well. So there's 4 possible genotype combinations for the progeny of this mom and this dad, right? So let's imagine mom's egg is haploid G and dad's sperm is haploid A. That would lead to a heterozygous AG genotype in the offspring. So would the reciprocal of that. So the probability of an AA genotype is 1 out of 4. There are 4 possible genotypes. The probability of a heterozygous genotype is 2 out of 4. And the probability of that last genotype, GG, is 1.4. Now, this is a very simple framework. We can think about this intuitively. We were taught this in high school and in college and now in grad school again, and it's probably something many of you think about a lot. And it's intuitive. It's just, we just know this. But it's actually, it is a random, there's uncertainty. And this is a model, this is a probabilistic model for measuring or modeling the uncertainty about the frequencies of different genotypes in offspring. And it's an expectation, right? So if we have a mouse litter, this is a mom mouse and a dad mouse, and they have a bunch of offspring, we expect among those offspring that the proportions of these different genotypes will be 1 to 2 to 1, right? 1/4, 1/2, 1/4. It's not always exactly that, right? Why? Why isn't it always exactly that? Assuming there's no sort of biological biases. Well, what if you have 2 offspring? Yeah.
They're independent.
They're independent trials, but also you need to actually get exactly those proportions. You actually need a lot of offspring, right, to actually see exactly that. So if you had 3 offspring, you cannot see those proportions, right? So it's sort of the model of how we think about the proportions, but you actually need a boatload of progeny to see exactly, even anything close to exactly those proportions. So this is what probability theory 4 is really to help us model, and I think in the context of publishing results and thinking about, you know, what goes in your discussion section, like what's potentially wrong with this, what's the future work. It's really about the uncertainty about what you saw. Here's the data, we interpreted the best we could, we modeled uncertainty, but there's actually follow-up work because there's uncertainty we can't account for. It's usually what we're talking about. Okay, so discrete random variables, as I mentioned before, they're used to model processes where there's a finite set of outcomes. So I mentioned, so an example would be coin flips. You've probably heard of the binomial distribution. Who's heard of the Poisson distribution? Yeah, so that's another discrete one. It helps you model count data, reads aligning to the genome, for instance. So some discrete random processes or variables would be sort of how many goals in a particular soccer match. One of the reasons soccer is not terribly popular in the US is often that number is 0 or 1, so people get bored. But it's 1 or 2 or 3 or often 0. Did a mutation occur? So in this case, it's a binary, but there's two outcomes. There's a yes or no. It's just like, was there a mutation that's cell division or not? That's a random process, a discrete random process. The number of transcripts for a given gene in a given cell at a given moment in time, right? That's a dynamic process. Sometimes it's responding to exogenous or endogenous stimuli, or it's just randomness. You know, polymerase is binding. RNA polymerase is binding and transcribing and firing off. Sometimes there's a lot present in the cell, sometimes there's a few. And when we do a big scoop of cells for RNA-seq, we're essentially taking an average of across all those cells. So if it's variable from cell to cell, we sort of get a sense of what the frequency of that molecule is across a population of cells. I talked about this already, possible genotypes, as I just showed. All right. So let's talk about something that really confused me a lot when I started working on these problems. There's this notion of a Bernoulli trial and the binomial process. And I don't know if any of you have worked on this. We're going to sort of talk about Bernoulli trials and binomial processes, and hopefully I'll clear up some of the confusion that plagued me for a while. So a Bernoulli trial is this. The name comes from this mathematician whose last name was Bernoulli. I don't know much else about him, but essentially he formalized thinking about experiments where there's exactly two possible outcomes. Okay. So what's an example of two possible outcomes? Well, flipping a coin is sort of the most common example. So we know it as heads or tails. I guess in China a long time ago, it was named as ship and head because the coin that was used had a head on one side and a ship on the other. And for reasons that are unclear to me, in England a few hundred years ago, it was known as pile and cross. I'm guessing for the same reason. I've never seen that coin. Similarly, if an individual is heterozygous, A or G, you can think of this, you can sort of generalize that to a heads or tails problem. So if an individual is diploid at a given locus and heterozygous, AG, and you're studying RNA-seq molecules from either the A or the G allele, say mom and dad's allele, every read is either A or G, heads or tails, right? You can think about more complicated scenarios where, you know, a deck of cards has, 52 cards in it. There's 4 different suits, 13 cards per suit. So it's hard to maybe think of that as a Bernoulli process, but you can turn it into simple binary questions. If you shuffle a deck, draw the top card, is it an ace? Right? What's the probability of it being an ace if you shuffle the card? If it's completely shuffled, pile up, draw the card. Anyone want to guess? How many aces are there? How many cards are there? Right, so it's 4 over 52, which is what? Right. Right. So the probability, if someone's really done a good job shuffling, if you play gin or something like that, and then try to pull the top card, maybe the probability is a bit different. What was the probability? What's the probability of rolling a 6? Fair die. 1 out of 6, right? Okay. That is not a Bernoulli trial, but if we turn it into what was a 6 rolled, yes or no, that is a Bernoulli process or Bernoulli trial. So we're gonna do some experiments now based upon coins. So imagine a fair coin. The probability of landing on a heads obviously is 0.5. The probability of landing on tails is also 0.5, or 1 minus the probability of heads. And the important thing here is that the sum of those two probabilities equals 1. And what does 1 actually represent here? 1 represents all possible outcomes. So this might be slightly flawed, like I can't imagine how this would ever happen, but imagine you flip a coin and it lands exactly on the side and stands straight up. That actually wouldn't be modeled by this. So maybe it's like, this is 0.049999, and that's 0.499999, and the probability of landing on the side is 1 minus those two 0.499 things. But I think for these purposes, let's assume those are the only two possible outcomes. All right. So the expected frequency of heads, if you flip coins, is 0.5, right? Does that make sense? So if we flip a coin a whole bunch of times, we expect that half of those flips will be heads. But really, I think this is getting into sample sizes, right? So if you flip a coin just once, can the expected frequency of heads be 0.5? Or the observed frequency, I'm sorry. No, right? It's either yes, 1, it was a heads, or 0, no, it wasn't a heads, it was tails. Right? So even though that's our expectation, the expected frequency of heads is 0.5, we only can observe that if we do lots of coin flips. And that's analogous to the problem of, the expected genotypes in a mouse cross. You only get to see really close to those proportions, 1 to 2 to 1, if there's, say, 10 or 20 offspring. So Alan will be formalizing this a little bit more, but you're going to hear about the expectation or the expected value for a given process. And that really comes from the expected number of heads here. If the probability of heads is 0.5, the expected number or frequency of heads is the number of flips times that process, or times that probability, right? So the expected number of heads is just n times p. So for example, if we expect 5 heads in 10 throws because 10 times 0.5 equals 5, right? But what if we did 3 throws? 3 times 0.5 is 1.5, which is actually a value we can't observe. So that gets at the difference between what we can observe in small sample sizes and what the expectation is. And that's a shorthand way of thinking about, that's why we want lots of observations, lots of samples for biological processes, because when we have low counts or low trials, low depth for an RNA-seq experiment, for instance, there can be huge differences between what we observe and the actual truth, the expectation for the process. Okay, so there's a difference. There's one concept that fairly simple, but there's frequency of an event and the relative frequency. And these two are often conflated, and you can see this in a lot of figure legends. Y-axis is usually labeled as frequency, when actually what it is relative frequency, or vice versa. So what are the differences? If we flip a coin 10 times, the frequency of heads is, say, the expected frequency is 5. Right? We flip 10 times, we expect to see 5 heads, but the relative frequency is that observed value over the total. So the proportion of the events that met that expectation. Okay. All right. So we're going to do this. We're gonna demonstrate why this can be a problem. I brought some pennies. So does everyone have access to the slides? If you don't, go to the GitHub site and you can access the slides, which will then allow you to access this. But it's pretty simple. It's just bit.ly/slobs-coins. So what I want everyone to do, if you could take a penny and just hand roll back. So if you have a coin and you have the spreadsheet open, you can get going. There's 3 columns in the spreadsheet. So I'm going to open up this spreadsheet. Oh, wait, can I have a penny on it? So if you open the spreadsheet, oops, I put some toy data in there. Let me clear that out. So put your user ID in and do 10 flips and track the number of heads. So this last column should be 10 for everybody. It's kind of redundant. All right. All right. It's actually kind of hard to keep track of. After you do a few, you kind of forget. Has anyone finished? Has anyone rolled all 10 heads? Whoa, really? 10? 10 heads?
No.
Oh, 7. Did anyone get exactly 10 heads? Did anyone get 0? Good. A couple people finishing up. Did anyone get just 1 head? Wow. Did anyone get 9 heads? Anyone get 2 heads?
Yeah.
Okay. Just 1? 2? You did? 2 heads? Okay. So let's look at this. So there are 10 flips. I think I got these from the bank, so I think we can assume they're fair coins. So what's the expected number of heads? We've already talked about this. 1? 5, right? So looking through this, there's a few 5s, but not many. Most are sort of close to 5, but not quite. A lot of 6s, a lot of 4s, a couple 7s, a couple 3s. Apparently no 1s. All right, now so I'm gonna clean this up a little bit. Looks like there's some stuff we gotta clean up. Right. So what I want to do is play around with this data a little bit to see what's going on with it and see. Okay, there's a couple of people that said they did 4 flips. I think that's a lie, right? Did you actually do 10? Oh, I see. Someone put 6 and the remainder is 4. Okay, we'll do this. Boom. Did this person use 6020412 actually do 20 flips and only got 2 heads? Who is that? 10, thank you. Just checking, sanely checking our data. Let's see. Anything else that looks wonky? Oh, 4 and 6. There we go. Fix that. Fix this. Delete some rows. I think I butchered that. Sorry. Right. Does anyone else see anything weird that I should clean up? I just did a quick scan here. Looks like no one had 11, which is good. All right. Okay, we're in good shape. So that was an experiment. Okay, you guys did a bunch of Bernoulli trials. And essentially, each of those independent experiments were samples of this random process. And what I hope, I kind of did this a few different ways with my own simulated data, and I'm hoping it illustrates a basic point. We'll see. If not, I'll have to sort of riff it a little bit. So do these results match our expectation? I think we can obviously see no, because not every experiment was 5. But what we can actually ask is a more nuanced question, which is, does it match our expectation given low samples? I mean, we know that we've only done a few coin flips, and we know, for instance, that I think we have some intuition that when you only do the experiment a couple times, say once, we're never going to get 0.5. If we do it twice, there's only one scenario where we can get 0.5. If we hit head or two scenarios, heads then tails or tails then heads. But if we did heads heads or tails tails, we wouldn't get 0.5. With 3 coin flips, we can never get 0.5, right? With 4, we can. With 8, it's much more likely they would get something really close. With 20, with 50, with 100, with 1,000, we should get even closer to 0.5. Okay, so we are gonna, I'm gonna teach you a couple different things with R. So now we're gonna do some coding. We are going to install this data pasta package, which is useful for situations like this, where we've got a little data set that's either up on a HTML page in a nice table or a spreadsheet, and we just want to paste it into R very quickly because we have some code that we know will work. We want to plot it or we want to do a statistical test on it. So the way, I'm going to have to get out of the slide here. So you should be able to just, we're just installing a new package as we've done many times before now, and then we're going to load up that package. I'm going to kind of blow through this because you can refer back to the documentation for it later. But essentially, the name comes from, it's not pasta, I guess. It's, I guess, Canadians say it's sort of similar, like pasta, but I think it's mean like data pasta. It's a paster for data. I'm guessing. Oops. Okay. Hold on. Yeah, there we go. I broke my R session, but I think I'm good now. Okay, so it's installing it, and then I need to load it. Data pasta. Okay, so here's the thing. When you do that, you get this cool button up here. Well, it probably already existed, but there's something called add-ins, and it adds this new section from data pasta, which allows you to paste data as data frames or tribbles, tibbles or tribbles? Tribbles. As Charlie talked about last time. So let me show you what we do. It's really simple. As you can see in the second step, we copy that range of data from the spreadsheet. So I go up to the upper left-hand corner, oops, and I can drag all the way down. Probably see a bunch of people doing this. So we've got the whole thing. I'm anonymous chameleon. I have that selected. I can hit Command-C on my computer to copy it. Then I can go over here, go into this code section, go to add-ins, paste as tribble, and check it out. It creates all the code to load that up as a tribble. Okay? So we're not going to run that yet, but the next step is to, after we've pasted in that code, we need to make a variable out of it. Is everyone with me? I'm moving kind of fast intentionally, but hopefully the slides are good enough that you can catch up. So I'm going to take that pasted code and assign it to a variable, a tribble, a data frame, essentially a fancy data frame, called coin flips. Okay? So up here, I'm going to say coin underscore flips equals that whole tribble. I'm going to select all that code with Control A, and I'm going to run it with Command Enter. Okay? Now, if I say coin flips, I get the whole, it's only showing me the first 10 rows, but apparently there were 43 people in this room that participated in the experiment, so we've got 43 data points to look at. Okay? Is anyone having crazy trouble, or is it going okay? All right. I'm going to move on then. So we got a data frame that's basically all the experiments that we did on the Google Sheets, and we use data pasta to do that. Right, so we can now use ggplot to just make a plot of the number of heads divided by the number of flips, which is what? It's not the frequency, that's the relative frequency, right? The total number of observations over the total number of experiments. So in other words, like going back to that weather example of 0.70% chance of rain, you know, that's basically, they did 1,000, they took 1,000 data points and asked for how many of those, under the exact same conditions, how many times did it lead to rain in the past, and we'll use that as our prediction for Okay, so we're going to use ggplot, cowplot, which makes it prettier. If you remember, I think it's cow is the initials for the guy that created it, Klaus Wilk. I don't know what his middle name is, but it starts with an O. And then we use ggplot. We tell it the data frame that we're using. Remember, we always have to give it an aesthetic. The X here is we're going to automatically divide the number of heads that we saw, which is the second column in our spreadsheet, by the total number of flips, num flips, and we're going to make that into a bar chart. Okay, so you can just copy and paste that guy right in here, and you should see something that looks like that. So your experiment looks much nicer than mine because I just did I think I got bored after maybe 10 different tries of this. So we can take this. I'm going to update the slide. Wait, sorry. Copy to clipboard, copy plot, ditch this, put this in there. Okay, so where's 0.5? Well, 0.5 is right here. We had a bunch of experiments that were between 0.4 and 0.6, so it wasn't quite the most commonly observed relative frequency, but it was pretty darn close. It looks like the most commonly observed relative frequency was 0.6. So maybe the bank gave me some slightly biased coins, probably not. It's just the randomness of the experiment. It's the uncertainty about the result with only 10 flips. Okay? So where I'm working towards here, and I think this is probably intuitive, is what if we had done 20 flips? What would this have looked? What would you guess this would look like if we did 20 flips? Do you think this bar, 0.5, our expected relative frequency would be the highest? Maybe, maybe not. I would guess it would be 2, but it might be that it's still not the highest after 20. But I would bet you if we all did 100, that'd take the whole hour probably. But if we did 100, we probably would see this nice peak right at 0.5. Okay. Any questions about that? Hopefully that makes some intuitive sense. So why are there so many outliers? Yeah. Okay, on slide 16, step 4 has the code that you paste in. All you're really doing is taking this little bit here, coin flips, and basically taking that triple and assigning it with this left operator to coin flips. Okay? So why are there so many outliers? I think I've beaten this horse to death, but one of the fundamental concepts in probability is that with fewer trials, for instance, flips, you expect to see a larger difference between the observed relative frequency and the expected relative frequency. I've sort of enumerated cases like that. You flip it once, or flip it twice, or flip it 3 times, you really can't get the expected relative frequency. But the corollary is, and this is important for biology, is that if we do more trials, we get deeper coverage from RNA-seq, or deeper coverage from DNA-seq, or ChIP-seq, or whatever you do. The difference between the observed frequencies, relative frequencies, and the expected reduces. So you get closer and closer to the truth with larger sample sizes. Okay? And let's just see this with simulating some coin flips in R. I'm going to leave this code up here for you. I'm going to go through this quickly just to demonstrate the point, but I encourage you to revisit the code. Essentially what we're doing, so this is the disconnect between Bernoulli and binomial. So I'm saying I want to simulate 10 Bernoulli flips of a fair coin. But in R, we do that with this thing called R for random. It's generating random results from, I would have expected to be R Bernoulli, but it's actually called R binom for binomial. So I'll come back to that in just a second. But what we do is we're asking it to flip it 10 times. That's this first parameter. And each flip represents one experiment. And we're saying that the probability of flipping, of success in this case, is the probability of the heads. So essentially 0 means tails, success is 1 means heads. So in this case, it just did random coin flips for us. The computer did it. And it looks like 3 out of the 10 flips were heads. 1, 2, 3. Okay? So the note here is that Bernoulli trials with more than one trial, 2 or more, are generalized as with the binomial process. So Bernoulli trials are restricted to just 1 flip. Okay, so Bernoulli is sort of binomial is a superset of binomial. So there's a whole bunch of functions in R that start with R for random and then have the name of the distribution of the name of the random process from which the random trials will be generated. So R binom basically means it samples random numbers from a binomial distribution with this probability, with the probability of success. So coin flips, if we wanted to model, let's say we put some lead tape on the head side, and maybe, don't know if that would bias you towards tails being up or heads being up, but that biases the coin in some way. So you could reflect that by changing the probability of heads or tails to be, instead of 0.5, make it 0.6 or 0.7 to show that it's a biased coin. Okay, so we can use this code to do really, really big experiments, just like we did in class by flipping coins, but in like a half a second, we can simulate 1,000 coin flips and just see what happens. So what we're going to try and show is that as you do the flips more and more, if you flip more and more times, the relative frequency that you observe of heads is closer to what you expect at 0.5. So let me just show you a few examples. Bear with me here. So for instance, if we do 1 flip, this time I got a heads, next time I got a tails, we can just keep doing this over and over again. This is a Bernoulli trial, just 1 flip at a time. Okay, I'm going to fast forward, and this will tell us actually the result is not the 0s and 1s of the result, it just gives us a count of how many successes there were. So this allows us to quickly do a whole bunch of coin flips and just get a number back that tells us how many were heads. And that allows us to really quickly compute this relative frequency. So for instance, the last time I ran 100 flips, I got 44 heads. Let's see what I get this time. 54. So we're going to ask the computer to flip 100 coins again. It does it way faster than we can. 44. 50. Ooh, that's a good one. Exactly the expected frequency, right? Okay. So this version of the R binomial function allows us to do lots of flips and just have it return how many of those flips were successes, or heads in our case. All right, so this series of code is a big simulation of this process. So I'm going to create a data frame or a tibble, and I'm going to create 1 column in it that has between 1 and 2,000 flips that are going to be tried. So the first row is the first experiment where we're just going to do 1 flip, and then it skips, it adds 2. So the next row will be 3. The seek 1 to 2,000 by 2 means start at 1, then go to 3, then 5, then 7, then 9, all the way up to 2,000. So each of those rows, this num flips column is going to be the number of flips that we want the computer to do. So essentially the last row will be, we're asking it to do, I guess will be odd, 1,999 flips, and tell us how many of those were heads. Okay? So I'll just show you what that looks like. Right, oh, I got to import tidyverse, sorry. To get this to work, you'll have to say library Let's see if that works. Okay, so we've got this data frame called coin flip data frame. And look, it just has that number of flips, 1, 3, 5, 7, 9, etc. Okay? Now what we're going to do, this is some nasty code, I got to say, but I'm going to walk you through it. Remember Charlie last time, if you were here, talked about deep liar. If you weren't here, this is going to be confusing, I apologize. But you take that coin flip data frame. Let me use my cursor here. You take that coin flip data frame and you pipe it, the contents of it into this function called rowwise. And that means for every row, I want it to do a particular thing. And that particular thing is right here. We want it to mutate the data frame by adding a new column called numHeads, which is the result of calling that random binomial function. I want it to tell me how many heads there were based upon the number of flips that are in the num flips column for that row. So it's starting at 1, it's going to do 1 flip, and it's going to return. The contents of this num heads column will now be, for row 1, how many heads were returned when we did 1 flip. Then for the next row, there's 3 flips. It'll populate that num heads column with how many heads there were there, and so on and so forth, all the way to 1999. Now, the one trick here is the reason we have to do row-wise is without that, it'll take the last, the number of flips from the last row in the table and use that as the number of flips it does for every single row in the table. So it'll be basically the whole table data frame will be populated with the results from flipping 1999 times. So instead we want it to do it row by row or row-wise. Okay? So let's see what that looks like. Okay, that was really fast. So it just did 1999 plus 1997 plus 1995 all the way down to 1 flips in like the blink of an eye, right? Isn't literally flipping a coin, but you know what I mean. Okay, so now we see our 2 columns. This is the number of flips, and this is the number of heads that were observed for those flips. And you can see it's, relative frequency for the first one is 0, the second is 0.33, the third one is 0.4, the third one is, I don't know what 3 divided by 7 is, but something just under half. Okay, so now we've built up this data frame where we've done a whole bunch of experiments. We've got the number of flips and the number of heads observed. Now we can just automatically divide number of heads divided by number of flips to get that relative frequency. Okay, so that's what we do here. We're just adding a new column called relative frequency or relfreq. And that's just obviously dividing one column by the other. Not sure what I did there. So let's take a look at that. And now we get this relative frequency for every one of them. So it turns out 3 divided by 7 is 0.429. Okay, so then this is just using ggplot again to essentially plot as a function of the number of flips the relative frequency that was observed. Right. So what I've drawn with this red line is our expected relative frequency at 0.5. And you can see that it oscillates when we have just a few number of flips. It oscillates all over the place around 0.5. Sometimes it's like 0.1, sometimes 0.9, etc. And then it starts to converge. I mean, there's noise, but if you compare the relative amount of noise way out here with 1999 flips versus 2 flips, you can see with many, many trials that the observed relative frequency converges to very close to the expected relative frequency. Okay? So that introduces something called the law of large numbers, which I think is something that's one of the more intuitive concepts in probability, but it also gets misused in some ways. But let me just rip through this. The idea is that, obviously, as we just showed, results obtained from large numbers of trials should be closer to the expected value and will tend to become even closer as ever more trials are done. So if we did this all the way out to 1 million, I would guess that we're going to be really, really close to like 499,999 heads or 500,000 or 500,001. So if you compare that that extra one or that missing one relative to the total number of trials, that proportion is really small. So 1 out of 1 million is so that variance, we'll talk about variance more formally later, that's essentially the variance of the experiment, and that variance gets smaller and smaller as we do. So basically we're visualizing variance here. It gets smaller and smaller as we do bigger and bigger experiments. And in biology, the problem we're often faced with is that we kind of know that we need to do these bigger experiments to have better data, but there's cost involved, there's money. So it's this balance of doing the best experiment we can with the money that we have, and then being able to model the consequences of just doing that smaller experiment with the money or the technique that we have. Okay, I just want to touch quickly upon this guy, John Kerrich, because the experiment we just did is something that he actually did himself manually while in jail in Denmark in 1940. He was going to visit his in-laws in Denmark, and the Nazis came into Denmark, and he was interned as a consequence of being in the wrong place at the wrong time. And he and his buddy Erik Christensen basically were clearly bored to death or just very curious, and used coins and fair coins, and they biased coins in different ways and used ping pong balls for reasons that we won't get into, but basically to demonstrate empirically so that some of these fundamental concepts in probability theory actually bear out when you do big experiments. And literally, they did this experiment all the way out to 2,000 coin flips. And someone just sat there and wrote it down and flipped, and wrote it down and flipped. And this is actually an excerpt of their data that's been converted into a text file, but it's essentially from like scanning their zeros and ones when they wrote down the experiment on paper, and then they scanned it in, converted it to text, and this is a snippet of it. Pretty crazy. Okay, so in the last couple minutes that I have, I'm just going to touch upon two quick things. I've already mentioned the fact that coin flipping, has a heads or a tail, a yes or no. It's very analogous, or it's a great way, a conceptual framework for thinking about the expression of two different alleles for a gene, right? If we know that, we know that we inherit a gene copy from mom, a gene copy from dad, and let's say we inherit in that gene an A from mom and an A from dad, or a G from dad. We can actually empirically measure how often the A allele from mom is expressed versus the G allele from dad. And we expect that it's 50-50. That's kind of what we're taught. But RNA-seq allows us to actually directly measure that. But sometimes RNA-seq, and so A and G is essentially equivalent to heads or tails in this context. RNA-seq, that model or that observation of mom's expression versus dad's expression will be fairly informative if we have an unbiased experimental condition, and we have large enough counts where the relative ratio of A and G is kind of what we expect. Our expectation here is 0.5. Half of the reads will come from mom. Let's say mom is success. That's probably the right way to think about it. But if we just have 2 read counts, it's not terribly informative. Okay, so that's the connection. And so, if we have biallelic expression, we'd expect this sort of 0.5 from mom, 1 minus 0.5 from dad, so this equivalent heads or tails. But if there was some bias coin, let's say blue is mom, that's heads, that it's not a 0.5 expectation. There's sort of like 0.8 expectation for mom's allele. And then we could have one allele completely shut off. And if we took the RNA-seq data and essentially plotted these relative frequencies as bar plots, these are the ratios that we would expect to see. The whole point of this is to show you that this is not too far removed, or actually not removed at all, from the concept of coin flipping. So the reason we went through all this coin flipping experiments is really because it's a fundamental way of thinking about a lot of different biological counting experiments, where we have 2 alleles or 2 states, and we're basically counting data from genomic experiments to measure the 2. All right, I've run out of time, so this is stuff that Alan's going to revisit anyway next time, so I'm going to skip ahead and just show you. You can go look at these later. There are two things I would really recommend reading before the next lecture, so that'll be in 2 weeks. One is the first 3 sections. This is literally like a page and a half from Alan's really well done white paper called Just Enough Probability. He teaches this down in anthropology every year, and he's been kind enough to come up and help me and others with some of the teaching. So the series that I was talking about is called Points of Significance. There's this first paper called The Importance of Being Uncertain. It's a 2-page, very accessible, I think, discussion of fundamental concepts of uncertainty and probability theory. It's really worth your time. The whole series is worth your time. I've now read the whole thing and kind of blown away by how great it is. And then there's 2 questions that I won't take the time to read here, but it's a way to synthesize the work that we did today and thinking about how it can be connected to biological experimentation. It's 2 fairly simple thought experiments. If you have time to work on them, great. If not, that's okay too. Alan's probably going to revisit these topics next time, but it'll help set you up for the next lecture if you maybe think through these a little bit. So next time he'll talk about combinations and permutations, which gets to the slides that I skipped at the end there, the law of independence and the multiplication rule. And then he's going to talk about, on the 19th of August, conditional probability and Bayes rule, which is incredibly useful. And then I think September 5th, I'm going to start talking about more discrete random variables, the Poisson distribution, expectation and variance and things like that, and how we connect it back to counting experiments in biology. So that's it. Thank you.